I was an awardee of the Rising Star Award from what was then IMRO, what is now One Mind, but really from the Staglin family. And so I owe them an immense debt of gratitude, Sherry and Garen. You supported me throughout my career, and I really appreciate that. And the Rising Star Award was an opportunity to, yes, meet wonderful scientists, and it also was a key contributor to the ability of me to expand the lab at the time as I was a young investigator and really do some wonderful work. I'll hint at some of that work today, but really what I want to tell you more about today is the overall state of mental health research as uniquely I see it as a director, which means, of course, I'm going to give you information that's probably wrong and ideas that are probably inaccurate, according to many of the folks in the first few rows here. But at least it'll start a dialogue about what I think of the challenges and opportunities in mental health research. And I should also acknowledge Tom Insel, who's in the audience, my predecessor at the NIMH, for many reasons. One, he also was very supportive of my career throughout that career and helped me get that career going. And also because he left an institute in such wonderful shape that I didn't have to worry about making sure the institute runs well, and I can think really just about science, which is what I love. So the mission of the NIMH should be clear to you all. It's to transform the understanding and treatment of mental illnesses through basic and clinical research, paving the way for prevention, recovery, and cure. And I really do, as we all at the Institute do, take this mission very seriously. However, to achieve that mission, we face fairly substantial challenges that are in some ways unique to psychiatry, although in some ways also true throughout medicine, but are often understated or dismissed. But I also feel like that there are amazing opportunities in mental health research due to where we are right now in the state of technologies around the brain and the state of understanding and acceptance of mental illness. And so I want to share with you those challenges and those opportunities and leave you with the message that you're present here because you're passionate about it. So I don't know if this message is necessary for you, but spread it around. Getting involved is really going to help us in the long run understand and treat mental illnesses better. So what challenges do we face in psychiatry? We face a number of them. First and foremost, of course, is the high burden of our illnesses, the illnesses that we face. The second is the complexity of those illnesses. And I'm going to talk a little bit about the complexity in particular about the state of diagnoses in psychiatry. The third challenge we face is we have no lab tests that help us understand or track our illnesses. We have no x-rays that we can give to understand what's wrong with our patients and help guide treatment. And we call those things biomarkers because they're markers, if you will, of the biology of the disease, and that's what we're lacking in psychiatry. So that's a challenge. And then fourth, our treatments while they can be transformative for many, they are inadequate for way too many. So our treatments are overall inadequate. These are the challenges we face, and I'll go into a little bit more detail about those challenges. So the first challenge, of course, I mentioned is the burden, and this graph shows the prevalence of any mental illness in the United States, although these prevalences are pretty uniform throughout the world. At any given time, close to 20% of the population of the United States is suffering from a mental illness. That's slightly higher for females than males. You'll notice, though, in particular this third part over here, where we break it down by age, and you see that mental illness is most prevalent in adolescents and young adults. Mental illness is also present throughout any of the ethnic groups, and it really doesn't spare any of us. Although I think this is really interesting. 2 or more race or ethnic groups, and it almost doubles your rate of mental illness. So it means that the most vulnerable in our society are also the ones who are suffering the most. And what does this amount to? So if you have a mental illness, it is incredibly disruptive. I don't need to say it to this audience, but this audience may be surprised to know that in the United States, mental and substance use disorders are the third largest cause of disability in terms of life years lost, productive life years lost to illness. And in fact, it's number 1 worldwide now. So in the United States, it's number 3, and worldwide, it's number 1. Why are mental and substance use disorders so disruptive? It's because of that figure that I showed you just a moment ago, where it's striking young. So the other 2 major causes of disability are cardiovascular disease and cancer. And of course, those are diseases that tend, although not always, as we know, they tend to occur later in life. So you end up with a mental illness, and if it's not properly treated, if you don't achieve recovery, then you can be disabled for life. And that's one of the reasons why our disorders are so disabling. So that's the first challenge we face. We face a challenge of the burden of our diseases. The second is trying to understand those diseases, and the first step towards understanding psychiatric disorders is simply naming them, categorizing them, so that we can take individuals and group them together and study them. And this is a really challenging endeavor. The plot I have up here on this graph, these confused tangles of circles, represents the prevalence, represents the likelihood that an individual, in this case, child patient, presenting to a healthcare deliverer with a psychiatric problem. And each of these circles represents a diagnosis. And what you can see is that these circles overlap tremendously, because the rule for children, and I could have easily shown the same graph for adults, but I don't want to be too redundant here to waste your time. The rule is that you have multiple psychiatric diagnoses. Now, is it true that most individuals actually have multiple things wrong with their brain? No. What's true is that we don't understand how to categorize psychiatric diagnoses well enough to be able to figure out which ones are entities and which ones are simply descriptions of patients. And I'll get into that a little bit more later in the talk. One of the complications of diagnoses is what we call heterogeneity. And it's simply the idea that many of you may have observed that if you assign a diagnostic label to an individual who has a psychiatric disorder and the same label to another individual, they can actually have very, very different sets of symptoms. And as an example of that, I'm showing you in this schematic the diagnostic schema for post-traumatic stress disorder. For post-traumatic stress disorder, everyone starts out with some life-threatening stressor. And then once you've gone through that stressor, if you have PTSD, you develop symptoms in 4 clusters: re-experiencing symptoms, avoidance symptoms, symptoms of mood and cognition, and symptoms of heightened arousal. And each of those clusters of symptoms has lists underneath, because you can have heightened arousal in any number of different ways. And to get a diagnosis officially of PTSD, your doctor will go through and list all your symptoms and check them off these checklists and figure out if you have PTSD. And what this complex menu of symptoms means is that for the DSM-5, the latest definition of PTSD, there are 636,120 ways that a person can get PTSD. So there's tremendous heterogeneity. Now, sorry, you could say you laughed. I heard some laugh in the audience, and it is ridiculous that there are 636,000 ways. But really, really smart people have worked really, really hard to try to find better ways of categorizing people with PTSD, and they've failed. And it's not for lack of trying, and it's not for lack of being smart. It's for lack of understanding of the underlying brain conditions, and it's for lack of data. And so that's what we'll talk about later. So the first challenge, burden. Second challenge, diagnosis. Third challenge is biomarkers, right? So I sort of talked about this already briefly in the introduction, but when you go to the doctor and you have a fever and nausea and vomiting, the doctor's going to draw blood, right? And he's going to send the blood to the lab. And that lab is going to do a test on that blood. He's going to look for elevated white blood cell count, and or it's going to look for bacteria in your blood to see if you have a systemic infection that needs to be treated with antibiotics. And the doctor's going to categorize that bacteria to see which antibiotics the bacteria will likely respond to and choose her treatment based upon which of those bacteria you have. Alternatively, if you are suffering from dementia, your doctor now, actually already, will order a scan that measures the amount of amyloid plaque in your brain to help understand whether that dementia is an Alzheimer's type dementia or a dementia of some other type. Even though at this point, there's not a whole lot treatment-wise that you're going to do differently. But at least you can tell the patient what's causing the dementia, and you can give them some idea about what the subsequent course of the illness is going to be like. More importantly, from this perspective, this imaging biomarker in Alzheimer's, you can actually detect the presence of the amyloid plaques before the full-blown symptoms of Alzheimer's are there. So you can diagnose or predict a patient, and then once you do that, then you can work on strategies for preventing the onset of illness. So that's what you do if you go to a neurologist for dementia, or if you go to a primary care physician for an infection. What happens if you go to a psychiatrist? Well, they do a really good job of interviewing you, hopefully, if they're good, and they talk to you about your symptoms, and they do a treatment plan based on the symptoms, but there are no tests that they can offer you, right? They might send you for blood tests or a brain scan just to make sure it's not brain cancer, or it's not too much copper in your blood causing the psychosis. But beyond that, there's not a lot we can do to understand why you might be having psychotic symptoms, for example. So we need biomarkers. So we have high burden. We have trouble with specific specificity and heterogeneity in our diagnoses, and we don't have biomarkers to track the illness and help guide our treatments. The third thing is that our treatments themselves are of limited efficacy. This is a large study, a very definitive study that actually Tom and others at NIMH shepherded along of the efficacy of antidepressants in real-world settings. And they found out that if you put someone on an antidepressant, about 30% or so of those individuals will get well on that antidepressant. If they don't, you put them on a second antidepressant, another 20% or so. A third antidepressant, if they fail that, and you start getting diminishing returns. So that even with the best treatment, only about 70% of people, or slightly less than that, will actually get well on an antidepressant. So right away, we're missing 30% of the people who come in with depression. But what's not shown on here on the graph is that the majority of those 70%, if you wait out a year, they'll relapse. So even if you get well, you're going to get sick again, high likelihood, even with the best treatment. So our treatments, and this is true across the board for all categories of treatment in psychiatry. So our treatments, they do wonders for many people, but they don't help enough people, and they don't last long enough. And unfortunately, as many of you know, we see fewer and fewer truly novel drugs reaching the market in psychiatry. And we really need not just better treatments now, but a better pipeline for treatments that will provide those treatments in the future. Okay, so about now, you're feeling pretty hopeless. We have a huge problem, right? Very burdensome disease. We don't know how to categorize or describe that disease properly. We don't know how to track that disease properly, and we don't know how to treat that disease properly. Well, so why on earth would I go to Bethesda and take over the Institute of Mental Health that's charged with, improving this hopeless plot? Well, the reason is because there are really some amazing opportunities. And here is where I hope I can give you a little bit of stuff that you don't know so well. So, and there are opportunities in at least these three areas. I say at least because there are probably other ones that may be your favorite in the audience, but these three are the ones that I think about all the time. Number one, we have an increasing understanding of the genetic risk that leads to our disorders. And that provides opportunities for diagnosis, for understanding, and for treatment. We have an increased ability using really wonderful technologies, some of which you'll probably hear about today, and actually, which I based my career on visualizing and manipulating neural circuits in the brains of animals, and the hope is that one day we can bring that technology to humans. And then the third is an increasing understanding of and ability to use computational approaches to understand the brain and to understand our illnesses. So I want to talk to you about these three amazing opportunities to try to, boost the mood in the room a little bit before we go on to our great scientists. So what about genetics? So This is the state of genetics that I dreamed of when I was in graduate school, when I started working on mice and thought we would be able to use these mice to understand genetics of psychiatric disorders. There'd be some set of genes which cause or contribute to the psychiatric disease. And we could understand how those genes contribute to psychiatric disease by understanding the impact of those genes on the constituents of the building blocks in the nervous system, the cells of the nervous system, neurons, how those neurons hook up together to form the computational elements of the nervous system, neural circuits, how those circuits hook up together to form the vast systems of the brain that actually guide and produce behavior. And in fact, that's what I was telling you about some 8 or 10 years ago, I can't remember which, but Brandon might, when I received my Rising Star Award, where I wanted to look at a gene called 22q11, a microdeletion causing a syndrome called velocardiofacial or DeGeorge syndrome, and how it caused cognitive deficits by tracing its ability to influence the development of axons. These are the wires that go from one neuron to another, and the way those axons help form circuits and systems in the brain that guide cognitive behavior. And I was actually fairly successful in this rap, and I was successful because of the IMRO funding, among other things, and because of you out there in the audience. And I was also successful because I was really lucky, and that success led me to this position today. But in this position today, I realized that the reality is that the genetic picture of schizophrenia is way, way more complex than any one gene. So this is an image that some of you may have seen. It's actually now a few years old, from a landmark paper in Nature in 2014 that demonstrated that there are 108 places in the human genome that contribute to your risk for psychosis, that contribute to your risk for getting a diagnosis of schizophrenia. And now we know it's not 108 anymore. The last I heard was 180. I see Steve nodding his head. 180 is still the latest number, although I'm sure by next week it'll be 182. And so there's all these places throughout the genome that contribute to your risk for schizophrenia. And each one of them contributes a very, very small piece of risk. So that nice linear picture that I showed you before has been replaced by a much more complex picture. You have a bunch of genes. And those bunch of genes are paired with, of course, environmental effects, because it's not just about genes. And those genes contribute to multiple problems in the brain at the level of the neurons, at the level of the circuits, at the level of the systems that affect behavior. And this web needs to be unwound. And this might sound like I'm saying it's another challenge, but really it's another opportunity. Because more and more we're beginning to develop techniques that allow us to measure the effects of these genes on the brain in very, very high throughput ways. And it's my hope that over the next 5 or 10 years, you're going to see the outcomes of the studies we're funding now that really lead to an understanding of how this complex web works. And that we will begin to understand why that is, that schizophrenia is such a heterogeneous disease. And I mentioned schizophrenia because it's the first one where we have these hundreds of genes. But we're already starting to see returns in depression. We're already starting to see returns in bipolar disorder. And through a grant that IMRO is helping fund, the Aurora study that you're going to hear a little bit later, we hope to make the same inroads in PTSD, where we're just beginning to see the first few genes identified. So this complexity. It helps us understand why our diagnostic systems are so difficult. Because schizophrenia isn't one gene leading to one disease. It's hundreds of genes leading to hundreds of different phenotypes throughout the brain. But it also is the key we need, that genetic information, the key we need to unlock this complexity. Finally, the next thing I want to tell you about is neural circuits. So the brain, as I mentioned, is made up of neural systems. That is, these are large, far-flung entities of parts of the brain that work together to produce behavior. In turn, those neural systems are made up of neural circuits, which are, think about like an electronic circuit. They're nodes leading with wires in place, except the nodes are neurons and the wires are these axons. And so they're small connections of neurons that make a unified assemblage, a unified entity called a circuit. That circuit has one role to play in the brain, actually probably multiple, but we'll simplify. We'll say one role, which is to perform a computation. And that computation could be simply as simple as remembering what you ate for breakfast this morning, or as complex as predicting whether you're going to be happier in 10 years if you buy the red house as opposed to the green house, right? So that your brain is full of these circuits that are set up to make these computations, some simple, some complex. And the wonderful thing is we now have the technology to break open those circuits, look at them, and manipulate them to make hypotheses about how the brain works and to test those hypotheses. And these are technologies that many grantees from IMRO have developed and worked on, and which now we're seeing the returns of. So this, IMRO in one mind, I should say, too. So this is a picture of a particular part of the brain called the hippocampus, the dentate gyrus. And a technology developed by Jeff Lichtman and Tamily Weissman at Harvard University has enabled a multicolored map of this structure. And each neuron is actually flavored slightly differently. Each neuron is colored slightly differently so that you can tell which neuron is which in this morass. And actually, you can actually, not with this technology, with an additional technology, then watch that neuron be active as the animal thinks, right? And there are other technologies that allow us to implant molecules into those neurons and activate them, turn them on or turn them off at will, so that we can observe those neurons during behavior and figure out, this neuron is representing that you had a banana for breakfast, and this other one is active when instead of a banana, you have cereal for breakfast, right? And then we can test those hypotheses by, went too fast, by turning on that banana neuron and seeing if, this is all in animals, folks, right now, seeing if the mouse then presses the button for a banana, right? If the mouse knows that there's a banana in the environment because the banana neuron is active. And then if we can turn on the cereal neuron, then the animal will behave as if there's cereal in the room. So we have exquisite control. And just to give you a visual of the exquisite control, this paper actually uses a technology that allows you to pick out individual neurons in the brain of a behaving animal and turn them on or off at will at any given moment in time. And so they decided to be a little artistic and turn on a smiley face of neurons in the brain. And then what you see there is that image that lets you check whether that neuron's active or not. And you can see that these 6 neurons are active in the presence of a smiley face. Of course, we're not aiming to turn on a smiley face in the brain. We're aiming to turn on the neurons that will help restore function in someone with a deficit, or turn off neurons in a circuit that is causing problems. So for example, let's suppose that we have someone who has a psychotic disorder. If we could find and identify and characterize the neurons that are responsible for his or her hallucinations or delusions and turn them off, that would be tremendously powerful. So, of course, this presents another opportunity. We now have the technology to go and learn more about the brain and even intervene in the brain. And bringing these technologies translating these technologies into things that can actually be helpful in patients is one thing that we're really trying to gun for at NIMH. And in fact, we had a workshop just Monday and Tuesday bringing the best scientists really around the world who are working in this area together to try to figure out, okay, how can we transform our really amazing understanding of how mouse brain works into an even better understanding of how the human brain works. And then finally, we have computation. So most of us, myself included, probably avoided in math, although I suspect there's a bunch of you from the tech world here who didn't avoid math in school. But it turns out that math is really important, and that's one thing. Number two, I found in my postdoc when I had to use it, it's never too late to learn it. But why is math important for psychiatry? So it turns out for two reasons. One, we end up needing big data to solve that diagnostic problem, and I'm going to show you one example of that. The second, which I'm not going to talk as much about, it is if we really want to understand the brain, we need not just to be able to identify the neurons and watch them and then stimulate them and test our hypotheses. We need to have very, very firm quantitative hypotheses. We need to build what we call models of how the brain functions, and those models need to be mathematically accurate. So I'm going to not focus on that. I'm going to focus on the big data part, and I'm going to give you one example. Actually, this data comes from Connor Liston, who's a scientist in the audience, so excuse me for talking about your work today, but it's really wonderful, so I might as well. Where he studied the brains of 1,000+ individuals with depression and looked at how different parts of the brains talk to each other. And everybody's brain is slightly different, right? Everybody's brain, parts of your brain communicate to each other in different amounts. And so he created maps of each person's brain and the degree to which that brain is the parts of that brain are interconnected, are talking with each other. And then he took those 1,000 individuals with depression and asked, how similar is one person's brain to another person's brain in terms of how the different parts of the brain are talking to each other? And then clustered them, so put the most similar people together and the most different people farther apart, and found that within depression, there are basically, at least a loose approximation, 4 types of brains in depression. Now, that's not particularly interesting, except that then he asked, well, how are those people who now you've sorted based on how their brains are, how are they clinically? And he found really interesting clinical differences between these 4 types of brains. In fact, the people who own those brains respond differently to treatment. So one group of people with one type of brain responded better to a treatment called TMS than another group. He also found out that those people with these different types of brains tended to have different symptoms. So in other words, we can use these large data set computational approaches to try to figure out that heterogeneity in depression and make predictions about how an individual person will respond to treatment. This is a really promising avenue and one of the many reasons why we want more computation in psychiatry. Okay, so this last piece is a message that I know many of you in the room don't need, but it's to get involved. So, and why do I say that? Because if we're going to realize the potential of these great, amazing opportunities, and we're going to answer the challenge of these tremendous challenges that we face, we need everyone to participate in research. If we're going to be able to use computational approaches on big data, we need big data. And there are opportunities for individuals to participate in research, and that will really have an impact on mental health. And one of those opportunities is the NIH All of Us Research Program. This is the aim of this program, which started about 4 or 5 months ago and is gearing up to accept volunteer input by early next year, is to gather data from 1 million Americans who volunteer for their medical records to be anonymized and put in a publicly accessible database, who volunteer to participate in research, and who will allow us to look at and who will submit blood samples and saliva samples so that we can look at the connection between environmental effects, diseases, genetics. And what we want to do in psychiatry is take our measurements of behavior and disease and apply it to this group of 1 million people and use really large data approaches to ask whether we can do a better job of figuring out what kinds of illnesses there really are in the universe. And then be able to go back to those same individuals and say, hey, we want you to participate in this imaging study that's going to allow us to figure out how many people there are with depression, except it's probably not depression. It's really depression, bipolar schizophrenia. And we're going to be able to figure out within that group of people how many different types of people there are, considering their brains, their environment, their medical history, and their biology. And this requires that we as a community in the mental health advocacy community, that we get our folks who are suffering to sign up, that we get our folks who are suffering to participate in this research that is meant for them, that will hopefully identify better treatments and better understanding of mental illness. So with that urge, another thing you can do is you can learn about NIMH, and one good way to do it, if I could toot my own horn, is to I write messages nearly every month, sometimes multiple ones, that tell you what we're interested in. I'll be talking more about the All of Us research program. I'll be talking more about our efforts in computation, neural circuits, and other and genetics and other opportunities that we think we have at NIMH, and that's a great way to follow what's going on. And so with that, I'll just remind you about that mission, and I think I'm just a few minutes late, I apologize, but can take some questions now.